Well, I would like to start with welcome everybody and say good morning to also the audience. Hereby I open officially this academic ceremony in which Ms. Lidia Molina Millán will defend her academic thesis entitled Plastics and Their Effects on Aquatic Life Through the Lens of Mass Spectroscopy. May I invite you to present a summary of your studies and the conclusion of your thesis in the next 50 minutes, and I'd like to give you the word.
Thank you. Dear Prorector, dear committee members, supervisors, friends, family, and colleagues, thank you all for being here. And now I'm going to present my thesis entitled Plastics and Their Molecular Effects on Aquatic Life Through the Lens of Mass Spectrometry. I would like to start by talking about plastics. Plastics are present in our daily life, and that's because they have many advantages, and they are very persistent materials. And they are used actually to make all kinds of objects and for many applications. The only drawback is that the plastic waste is not disposed properly. So actually, there are huge quantities of plastic that end up in the environment, and more specifically in the aquatic ecosystems. That's why, in the recent years, scientists are trying to come up with sustainable solutions, and one of them is the bio-based polymers, and there are two types, the biodegradable and the non-degradable. And in this thesis, I will present one of these, and it's called polyethylene furanoate, or PEF. Even though there have been these attempts to be more sustainable, still plastic pollution is real. And microplastics play a key role on that. So microplastics are small particles of plastic up to 5 millimeters, and they are made of two main components. The first one is the polymers that comes from the fuel, and the second one, the additives. But where do these microplastics come from, actually? So they can already be made at this small size, or they can come through the degradation of the larger plastic items. And because these degradation processes are really different, we end up with a mixture of different microplastics that have different structures, different shapes, and different chemical compositions. In this thesis, we implemented a new technique to study plastics, microplastics, and their effects, and that technique is called Mass Spectrometry Imaging, or MSI. There are many modalities of MSI, but we decided to use two modalities. The first one, it's called matrix-assisted laser desorption ionization, or MALDI. And how does MALDI work? First of all, what we need to do is cut our sample into very thin slices. Then we place them into a slide. Then what we do is coat it with a matrix, that is a chemical that helps the molecules to come loose from the tissue. Then later on, we shoot the sample with a laser into very small spots. And then what this will do is make the molecules ionize and fly towards the detector. And after that, we obtain a spectra, and each of the signals in the spectra corresponds to these molecules that were flying. And because we were shooting in different positions, we are able to have chemical maps. So we know exactly where the molecules are located in the tissue. The other modality that we used, it's called secondary ion mass spectrometry, or SIMS. And this one, we don't need a matrix for coating, and instead of a laser, it uses a primary ion source. And it will work really similarly, and we will also obtain our molecules ionized and flying towards the detector. But SIMS is more focused to low molecular weight analytes or elements. And again, we obtain mass spectra and images. The overview for this thesis is as follows. I will present Chapter 2 that focuses on MALDI-2 MSI for polymer analysis, Chapter 3 on the synthesis and characterization of new copolymers, Chapter 4 on the acute effects and detection of microplastics in Daphnia magna. Chapter 5 on the chronic effects of foodborne microplastics in Dani orerio. And that would be it. So chapters 2 and 3 are focused on the polymers, and chapters 4 and 5 on the molecular effects on aquatic life. So let's start by chapter 2, which we implemented a new technique called MALDI-2. So this MALDI-2 works really similar to MALDI. We have the first laser, but we see that our molecules are not all ionized, so we want more molecules to be ionized. So what we do is shoot with a second laser that will intercept the primary plume, and then we will have more molecules ionized, so better results. The main question for this chapter was if MALDI-2 would work for polymers. So what we did, it was test multiple polymers, and we saw that for the polyethers and nylon 6, MALDI-2 compared to regular MALDI worked a bit better, but not so much. But then when we tested polystyrene, we saw a huge increase. And that's because polystyrene is able to absorb the MALDI-2 laser. You see it here in the graph, that there's a huge absorption band at the MALDI-2 laser wavelength. So then we move forward with polystyrene, and we tested in 3 different conditions. First, the polystyrene alone, the second, polystyrene with matrix, and third, polystyrene with matrix and salt. Salts are very used in polymer science because they help ionization. So let's go for polystyrene alone. Compared to the other ones, it's the lowest signal, but if we see MALDI-2 in orange compared to MALDI in blue, we see that there's an increasing signal. And in this case, because we don't have matrix, we call it laser desorption post-ionization. If we move on to the polystyrene with matrix, here we see a clear increase in the sensitivity. And again, this can be seen in the MSI images. The colors will work as follows. Really dark blue indicates low intensity, and yellow indicates really strong intensity. So MALDI-2 proves better results. And the last one, the third, we see that now because we added the salt with MALDI, we have some signal in blue, but still MALDI-2 works better. Now we move on to chapter 3 about copolymers. So we binded chemically 2 polymers, and the first one is PEF, polyethylene furanoate. And this polymer has very good thermal properties, and it's also 100% recyclable. But the only thing is that PEF is not really flexible. So what you need to do is bind it chemically with another polymer. And in this case, we bind it with PCl, or polycaprolactone. And this polymer gives the flexibility, and it's also biodegradable, which helps for the sustainable materials. The main goal for this chapter was to synthesize these new copolymers and analyze them in order to, in the future, have more sustainable materials that could substitute the conventional plastics. The workflow that we used is as follows. First, we synthesize the copolymers. Then we determine the physical properties. Later, we used MALDI-MS to study the structure. And to confirm that the synthesis was good. Then MALDI-MS to understand the structure. And finally, NMR to understand the microstructure. This was a collaborative project, so I would like to just focus on the MALDI results. So I will start with MALDI-MS. On top, you can see the spectra of PEF. So 1 unit of PEF would correspond to A, and this is in purple. And below, we can see the spectra of PCl, which is in red, and 1 unit of PCl would correspond to B. So we can see that we just have A or B signals, and that's because we have the pure polymers. But then when we synthesize the copolymers in the last spectra in green, we see signals containing A and B, which indicates that the synthesis was successful. Then we move forward with the copolymers to do MALDI-MS, and I will not go much into detail into the mechanisms or fragmentation patterns. I just wanted to show you that we obtained blockage structures. So how did we know that? Because we see different signals of PCl attached together, and the same for PCl. So these signals attached together indicate blocky structures of the copolymers. Now I move on to the microplastics in the aquatic organisms. So first we detected in Daphnia magna, and we used Daphnia because it's a widely used model in aquatic toxicology. It plays a key role in the food web. It's easy to keep in the lab, and it also presents advantages such as short life cycle, clonal reproduction, and high offspring production. The main question, or the goal for this project, was to implement a multimodal MSI strategy for microplastics detection and effects in Daphnia magna. So first, what we did is expose the Daphnia to microplastics for 24 hours, and we chose two microplastics, big and small, and they were fluorescent, because then we needed some reference in order to implement the MSI. So then we did the fluorescent microscopy to know where the microplastics are. And then we did the multimodal strategy, first SIMS for microplastic detection, and then MALDI for studying the effects. I will just discuss the MALDI results today. So for the MSI, we focus on two organs. The first is the gut, because we saw a high accumulation in the gut. So you can see in red highlighted the gut region of the Daphnia. And then we also focus on the eggs, again in red highlighted. And we focus also on the eggs because we suspected some reproduction effects. So now let's go to the results. What microplastics caused. First of all, in the gut, we saw an upregulation of methylene dialanine, which is a plastic additive. So that indicates that the plastic additives are causing toxicological effects in the gut. And for the eggs, we saw an upregulation of AMP and phospholipids, indicating that microplastics cause disruption in the egg development and the reproduction of Daphnia. And the last chapter, we wanted to know if we could also implement MALDI-MSI to study the local effects of foodborne chronic exposure of microplastics in zebrafish. So the animal model we used is the one as follows. First, we exposed the Daphnia again to these two microplastics. But in this case, we chose three different plastics: polyethylene, polystyrene, and polymethyl methacrylate, so 3 conditions in total. And then what we did is feed the fish with this contaminated Daphnia for a long period of time, 21 days, and then hopefully we would have some insights of how microplastics would impact human health. Now let's move on to the results. So in this case, we focus on the intestine of zebrafish because the fish were ingesting mainly the microplastics. So you can see highlighted in yellow the intestine, and also the MSI images show the intestine with a big accumulation of a bile acid called taurocholic acid. And comparing the intestine of the control and exposed zebrafish, we saw that the metabolites change, and the polystyrene was the group with most alterations. Now I will just show you some of the metabolites that changed due to the exposure. First, glutamic acid changed for all the groups, and we saw an upregulation. Let me show you as an example the control and the PMMA group. If we look a bit closer in the gastrointestinal tract, we can see that the control fish, the intestine is darker compared to the gut of the PMMA group. And that indicates the upregulation. So this upregulation might indicate a disruption of the gut microbiome, intestinal barrier, and immune system. Interestingly, for the groups of polyethylene and polystyrene, we saw an upregulation of energy-related metabolites, and these upregulations could indicate intestinal inflammation and inflammatory stress that are suffering the zebrafish. And lastly, because polystyrene was the group with most alterations, I also want to show two more compounds. So the first is the oxo1-in, and we saw again an upregulation. and this is a damaged DNA base. So this could indicate that we are having oxidative stress. And we also saw the upregulation of bisphenol S, which indicates again the importance of plastic additives and confirms the inflammation of the intestine. As a conclusion, today I've shown that MALDI-2 improves polymer detection, that MALDI enabled new copolymers characterization, and that MALDI-MSI revealed microplastics-induced localized effects in aquatic organisms. Thank you so much for listening, and I give the word back to the prorector.
Thank you very much, Ms. Candidate, for your very interesting presentation, well illustrated, and already my compliments before we're going to start. And we're going to start with introducing 5 members here today in this audience. First of all, myself, I'm the Professor Steinbusch, I'm the pro-rector, and I'm assisted today by Professor van Schoten, who is my secretary here. But more importantly, of course, is the promotion team on the other side of the table here. And I start with the first promoter, right on the other side of my chair here, is Professor Heren. He is professor in molecular imaging and assisted by Dr. Kuipers. She is not professor, she's associate professor in Mass Spectrometry imaging in peri-operative diagnostics, and assisted, that was Dr. Kuipers over there, and that professor's Dr. Silero Pastor. She is basically associate professor in spatial omics for regenerative medicine. And they have been helped you and guided you to do this thesis already in 3 years. Now, without further ado, I would like to speed up, and we're going to start the discussion. We have today for the audience 5 members in the Corona, and one of the members is online, and you will see the onliner later on the big screen. The opposition will be then opened by Professor Honing. He is also the chair of your assessment committee, and he is professor in systems imaging at our university, and I'd like to give him the first word.
Thank you, Mr. Pro-Rector. Dear candidate, first of all, congratulations with really trying to push analytical technologies in trying to understand pollution and plastics in environment. I read the thesis with a lot of interest, and I think there's a lot of promising information there. And also, of course, congratulate your supervisory team. But I would like to have some questions, and I would like to start on page 11. Actually, you say that MALDI-MS in the early stage of analyzing microplastic, nanoplastics. And then I looked to the reference, number 12, and I saw the year of 2020. And then I asked myself, well, I know that your own supervisor was already doing polymer characterization 35 years ago. So what's new in terms of early stage?
Hi, esteemed opponent. Thank you for your question and your kind words. So, okay, polymers, I would say they are old, but microplastics are quite recent. So there are few studies on microplastics.
Quite recent in our knowledge, but they were there, of course, plastics are already there for 100 years. So we are noticing them now recently.
So that's why we started the research recently, no?
Perfect. That's exactly what I wanted to hear. So it's the polymers are there, but focus on micro and nano plastics is new.
Yeah.
Clear. And then when you go to nanoplastics and you look to the definition of particles smaller than 1 micrometer, and then looking to the resolution in mass spectrometry imaging, let's say 5 micrometer, are you then able to see them?
That's a really good point. So actually, our instruments reach 5 micrometers, I would say. Well, the MALDI ones at least. TOF SIMs can reach lower. But yeah, I said nanoplastics are quite tricky. But you can, I think you could use another technology, more IR related, and those I know that they reach really like submicron.
Yeah, I think we will come back to that. Tip-enhanced Raman and tip-enhanced infrared would be also
nano IR,
yeah?
Those could also be an alternative if you think that it's not good enough.
I use the word promising because I think it's promising, but can I go back to proposition number 4 and ask one of your paranyms to read proposition number 4?
Yeah.
Please go to the microphone.
MALDI-2 enhances polystyrene detection, offering promise for microplastic analysis.
Now, the word promise means, yeah, maybe, I don't know, maybe not. But when I look to chapter 2, I see a lot of more than promises. I see a lot of steps forward. So why did you use the word promise in this sense?
Yeah, actually, there was quite a debate with my supervisor, Ron, but I think I'm less wild than him. So I just used promising because we didn't test it. Actually, the instrument was down, so I couldn't try it. But yeah, MALDI 2 showed really enhancement, so it's promising. But I couldn't test it, so that's why I chose that word.
If you can increase the resolution or increase the sensitivity for polystyrenes, and especially those are extremely difficult to ionize anyway, because they are aromatic, they don't have any functional groups, then you did an awful good job, because suddenly you see polymers which we did not see before. So why promising? I would even make it stronger and say we made the big step forward.
Yeah,
but you're reacting like, I don't know. Yeah. So I would say be a little bit more proud on that. And maybe have a little bit of discussion, because I start really to look to the mechanisms, of course. You say also in your presentation you are having a laser at 266 or 265, and then it's absorbing at the wavelength of, I would say, the aromatic ring of the polystyrene. But also in your matrix, you have the DHB, which is per definition also an aromatic ring. So is the chemistry which you explain us on, let's say, the page, whatever, the page, I will look to it, page 61, you have a nice explanation, of course, that multiple photons lead to the radical cation. That's absolutely what I understand. But I would say also the matrix is doing the same. So the chemistry happening there is much more complex, but also much more interesting.
But then, like, I don't understand what you mean. Like,
so in scheme number 1, you have a very simple, I would say, simple representation, because you say there's multiple photons at 260 or 266, and they will ionize the structure below. But the structure below is not the polystyrene.
No, yeah, that's the matrix. Yeah, that's the REMP for the matrix. Like, why do we see an increase on the protonated species? But for the polystyrene, it will be direct REMP. So the matrix is not involved in the ionization. It just helps the desorption.
Why not? Because if M is the matrix and the matrix gets a radical cation, the matrix is reactive.
But if you go to the page 65, to the figure 5b, if the matrix was involved, we would see something with MALDI, right?
Yeah.
But we don't see anything. So that's why it's not involved in the ionization.
Are you sure? Because you go from laser desorption to matrix laser desorption, and you see the result, but the result can be just protonation. And you explained us that there was a small change in MALDI 2 in terms of the ionization. Is that not due to the matrix then?
For the polyethylene glycol, yes. Yes, the matrix played a role in the ionization because of this REMP process. But for polystyrene, as I said, we think that the matrix is not involved in the ionization. It just helps the desorption, but the polystyrene ionizes alone. Well, it's direct REMP.
Okay, so I think we will continue that discussion.
Okay.
But very interesting and I would say more than promising technologies. Thank you, Mr. Prorector.
Thanks a lot.
Thank you very much. Then the opposition will be continued online by Professor Ognik. She is professor in environmental sciences at the Johan Stefan Institute in Slovenia. And I would like to welcome her and thank her that she joins us today all the way from Slovenia. And I'd like to give you the word.
Thank you very much, Mr. Prorector. And dear candidate, first I would like to congratulate you with your presentation. It was clear, well structured, and I really appreciate how your key accessibility and also engagement to this work that you show. So I have my questions are mostly related to the last two. chapters, and in your work with Daphnia and also zebrafish, how did you ensure that the observed metabolic changes really came from the microplastics and rather than from other, I would say, environmental issues that you have in your experiment?
Hi, esteemed opponent. Thank you for your question and your kind words. So the experiments that we did were really like lab control. So the particles that we chose actually, they have no additives. So it's like pure polymer, even though supervisors told me, yeah, Lidia, there's always a small percentage of additives, but we chose them like most pure as possible. So yeah, it's like we assume that like the control don't have any plastics and the exposed just have the microplastics. So it's an hypothesis that it comes from the microplastics. But what else can it be if they are treated the same way, just the microplastic differs?
Yes, but also you probably take higher concentrations of microplastics that you can see these changes, no? In zebrafish, usually, especially when I was working on silver particles, we actually use higher concentrations that we can see in the environment just to see how it was here the case as well, or
we use higher concentrations, like they are not environmentally relevant?
Yeah.
Yeah, we use higher concentrations because our focus was to develop a method. So of course we wanted to detect the particles. So we wanted to be sure that the particles were in the animal. So in that case we use higher concentrations. So yeah.
Okay. And then for my next question, which is related to this one, how the physiological differences between fish and the humans, how reliable are actually these models if you want to transfer the toxicology into the humans?
Yeah, that's a tricky one. So of course we share genes. Yeah, but we share genes with many things, like even with vegetables. So, but yeah, zebrafish is like we share organs, like we have brain, both of us. So I think like the effects could be translated, but of course there's always a gap in translational sciences. But I think that the effects we see in the zebrafish can give you an indication of what we can expect in humans. And that's really helpful for the regulatory agencies. So it won't be same as humans, but it's an indication of what to expect.
Exactly. This is very important that you can see some indications how this can be, how they can react, and might be also the case in the humans as well. Yeah. Because and then my last question is you was developing the MSI methods that how do you think that this will be involved in the environmental sciences in the future and in the next decades, specifically related to the emerging pollutants, because now this is the debate and also very hot topic in the community and scientific community.
So you mean that how it can be used in environmental sciences or other pollutants?
One of the
Sorry, and for many pollutants?
Maybe PFAS are one of the hot topics now. Do you think that this is then transferable, the methodology also to these ones or other emerging contaminants in the environment?
Definitely, it's on the earliest stages, MSI, but in my first chapter, like we did a review on the MSI in environmental sciences, and quite a lot of studies actually already started using the MSI, mainly for detecting where the microplastics are. And you mentioned the PFAS, and there are studies that already detected PFAS with MSI. So I think it's promising, not only for detecting the pollutant, it would also depend on the pollutant, but also to detect the effects. Like what I also shown, I think it's really useful to understand the effects at the local level. So I think it can give many insights on the mechanisms of action of the pollutants and help toxicology.
Okay, I have no further questions. Thank you very much, and good luck in your future career.
Okay, then we proceed, thank you very much, to Dr. Mengerink. He is affiliated with the Department of Polymer Chemistry and Mass Spectrometry at the Brightlight Camelot campus, and I also thank him that he joins us this morning, and I'd like to give him the word.
Thank you, Prorector. First, dear candidate, I would like to congratulate you with this really nice work. Really interested. I really enjoyed reading it. And I think you did an amazing job. I think also it truly adds something to society that we now, that we start evaluating this on this way. But my first response was actually, this can never work. You want to do multi-MS on microparticles, which are mainly polyethylene, polypropylene, they will never fly. And then at the end also, you know, if you want to look at toxicity, yeah, the most important thing is, what is the actual concentration? What is really, how much is it? Well, multi-MS is not known about his quantitation process. So, but yeah, when I started reading, you said, okay, I'm going to do TOF-SIMS on this. And I think, okay, that makes more sense to make TOF-SIMS for the polyethylene, polypropylene. But I'm wondering, how does that then compare to other technologies which you have? So, for instance, you have FTIR, you have pyrolysis GCMS, you have Raman, you have SEM, scanning electron microscopy. And they're all used for mapping MPs. And then you said, yeah, well, this technology, TOF-SIMS, overcomes the drawbacks of other techniques. On page 31 you said that, and the only thing what you say, you referenced it to reference 45. I looked reference 45 up. And then it said, yeah, for Raman, okay, you have some interruption with fluorescence of okay, I understand that. SEM, you say, well, it takes a lot of time, it's really expensive. I was wondering, is the TOF-SIMS, is that not expensive?
Hi, Steve Opponent. Thank you for your question and your kind words. So the last question, if it's TOF-SIMS not expensive?
In the comparison, you compare a lot of other technologies with TOF-SIMS on this page 31, and yeah, and you claim something like you claim for pyrolysis GCMS. Well, that is consuming my sample, but and SEMS I usually not use because it's too expensive. And then I was wondering, is TOF-SIMS not consuming your sample? And is TOF-SIMS, is that not expensive? So is that really the reason why to use TOF-SIMS?
Well, the reason why I used TOF-SIMS, it was because it was in our laboratory, so it was easy to access. But also it helped me to detection of microplastics. But I agree with you that for the quantification, MSI, it's tricky. For me, it's really difficult to imagine as an analytical chemist using MSI for quantification because you just have one slice, and okay, it can be representative, but it's really tricky. So, okay, if you have multiple slides of your sample, then you could try the quantification, but then that's really time-consuming, I would say. So for microplastics, I would suggest to the techniques you mentioned, and that's something that I didn't do in my PhD, and I think it would have been also promising, because in toxicology it's really important to know how much do you have, like the dose. So pyrolysis...
I will come back to the quantitation question later on, but I was just wondering how your picture is about all these other techniques, because you more or less in the thesis said, well, you have to do TOF-SIMS because all the other ones all have problems. But if I now hear you said, well, the other techniques can maybe also have some advantages stuff.
Yeah, like later on preparing for the defense, I was like, okay, maybe the other techniques were not so bad.
Okay, they're not line techniques, so I'm just wondering. And also on page 77, you compared also with NMR, and you say then, yeah, NMR is difficult for N groups. And again, I would say if you do TOF-SIMS on polyethylene, then I would say, well, N groups you will not measure. With NMR, actually, I think that is the technique of choice to look at N groups. Of, for instance, if you have branching of the polyethylene or no branching and these kinds of things. That typically you do with NMR.
Yeah, also with MALDI we could see the end groups, but it depends. Of course, if you want the polyethylene, polypropylene, forget about MALDI. But
yeah. Okay.
yeah.
And then my question is also, even if you would do MALDI in a complex polymer matrix, and then I'm talking about polymers, real polymers, and not what you just showed were oligomers with a mass of 1,000, but now I have a polymer which in real life are something 20 kilodaltons, 200,000 kilodaltons. Is then MALDI MS really such an easy technique to characterize your polymers in such a really difficult matrix?
You mean in the sample?
Yeah, if you have a tissue from a zebrafish or something like that. I never seen a zebrafish, so but I can imagine that you take a sample. There's all kinds of metabolites in there, all kinds of lipids are in there. For instance, if you would have polyethylene in such a sample, and you have lipids in the sample, do they not give exactly the same fragment in SIMS?
Yeah, could be, but the difference there would be that in my view, the polymer will have the really clear pattern. So for instance, polyethylene would be the difference CH2, 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 and I don't know how all the lipids look like, but I would not expect such a pattern with the lipids. So,
you're more an expert than I am on the topic, but would you not expect that you have a C1, C2, C3, C4 as fragments from a polyethylene after fragmentation, and you would have exactly the same if you use a lipid? Or would you not expect that if you have slip agents for like uricamide or other stuff that also have this long aliphatic chain, that they would give exactly the same fragments in your TOF SIMS?
Well, if in this case you could use other strategies such as the nano IR that maybe are more polymer oriented instead of the TOF SIMS that would give you.
But then you agree that these lipids can interfere with your observations?
Yeah, definitely we saw it, but I also thought that because our mass resolution was not at the best, I was like, okay, maybe if we, instead of TOF SIMS, we use the Orbi SIMS, that we get this higher mass resolution and we can distinguish these peaks. But if the high mass resolution is exactly the same, then we could use other strategies like ion mobility, for example. If the structures are different, then they will have different mobility, and that could also be an alternative to use.
Thanks. Then I had a question about quantitation, and in what I already mentioned, I thought you can never quantify this, but you came up with a reference for one of your colleagues here from M4I, that's I think Michiel van den Bosch, that he wrote, you can do it with an internal standard addition on the surface and then do quantitation. I think still, yeah, you have to exact the same compound to really do a correct quantitation. So recently I heard about a program that is called Pixis. I'm not sure if you heard about that program. That is a program which is based on artificial intelligence and machine learning, where you based on, and this is for metabolomics, so not for additives or polymers, but for metabolomics. But this kind of strategy, what they look at the mass spectrum and its fragmentation pattern, do machine learning, and with this program they claim that you can have a really accurate quantitation. I think within 15%. Well, but this is ridiculous good, I would say. Would you think that that would be an approach which you can also use for your quantitation problem, what you have? Because at the end of what I started my question with, this quantitation is really important. And so if you have too high or too low values, then yeah, you have an issue. But if it really has to be on a correct level. Do you think that these approaches would help you, or could help you, or maybe in future studies would be possible for your problems of these microparticles and additive quantitation?
So I think it could be a promising tool, but yeah, as we already discussed, like some microplastics are difficult to detect with MSI. So instead of the polymer itself, if we focus on the additives or the degradation products of these plastics, then I think that would be a more promising way to go. And as I said, like with MSI, like I think one section, even though it's really representative, you cannot quantify on that, in my opinion. So if you have multiple sections that represent the sample, then I think it would be a good way to quantify.
Okay. Thank you, and I would like to give the word back to the prorector.
Thank you very much. Then the opposition will be continued by Professor van Schoten. He's professor in genetic toxicology at our university, and I'd like to give him the opportunity to discuss with you.
Thank you very much. Compliments, dear candidate, with your work. It's very impressive, the technology, and also very relevant work. I really appreciate the interdisciplinary work. You must know something about a lot of things, toxicology, analytical chemistry, environmental sciences. So compliments with that, and also compliments to your supervisory team. My background is in toxicology, not ecotoxicology, but human toxicology. And I'm also interested in microplastics in humans. And it has been calculated that we absorb 1 credit card per week, orally or via inhalation, whether it's true, but it has been calculated. And also microplastic like polyethylene can be detected in blood, also placenta, and even placenta up to levels of 600-700 microgram per gram. So there's pretty much exposure to microplastic for humans. So my question would be, can your technology measure microplastics in humans? If, for instance, I give you a colon tissue sample or a placenta tissue sample?
Hi, esteemed opponent. Thank you for your question and your kind words. So your question is if we could be able to detect it with our technique?
Yeah, that would be great.
So we tested like really high concentrations that we focused on the molecular changes. So I would say in this case, like if it's a human sample, why not? I think we could also analyze it. But again, for the quantification part, I think still MSI it's a bit behind. So in the case of microplastics, I would use the pyrolysis GCMS for quantification. But then you would get this, you have bulk information. So then in that case, I think it's good to combine it with MSI. So because you have the bulk information with the pyrolysis GCMS to know how much and also what is it, like what plastic. But then with MSI, depends on the plastic, you can know where exactly it is and also what are the effects and like which part of the tissue is suffering the most.
Yeah, that would
so
be very
I think
important. Combining with pyrolysis GCMS and MSI, that would be a way forward. Okay. And can you also measure at a single cell level?
Yes, actually one of my supervisors is her research line. Okay. So I'm pretty sure you can do it with its challenges, but it's possible.
Okay, great. So environmentally, we are exposed to, let's say, environmentally changed plastics, which are aged or are weathered or whatever, and you buy your microplastics. So can your results, can that also, how comparable are your results if we want to translate that to the environmental exposures?
Yeah, so our experiments were like really controlled and like really pure microplastic, really pure polystyrene. So they are not that environmental relevant, I would say, but a way forward for future studies would be to do these aging processes such as thermal or even physical degradation that would be more representative of what's happening in the environment. And also adding these additives because in the end you don't have the just pure polymer. And also if they are in the environment, the microplastics, they can absorb other pollutants that are there. So it would also be interesting to study the microplastics and this absorption. Well, I'm sure there are studies already on that, but not with MSI at least.
Yeah, so it becomes very complicated.
Yeah, true.
Yeah, so we like, we are getting a kind of mixture toxicology and then see which compounds are responsible, which effects. Yeah, that's
would be tricky.
That's really tricky. I was also very interested in your chapters 4 and 5, of course, and also on the zebrafish model. And then on page 152, we have the figure. And I was very pleased to see that you can measure 8-oxo-DG, because that's also a DNA lesion which is really important for genetic toxicology. So I thought, wow, that's great. Because when we want to measure it, we isolate the cut tissue, the DNA, and then we measure it with analytical techniques. But you really can see it in the tissue. So I'm not that familiar with your technique. Can you explain me in simple words, how do you know it's 8-oxo-DG?
Sorry, this is the hay fever.
Yeah, okay, no point.
So how did I know it's this compound?
Yes.
So first we did the fast imaging, so the resolution was not, the mass resolution was not great. So then we confirmed the identity, so I had like a tentative identification, but to know that it's this compound exactly, I measure a consecutive section of the same fish, but with high mass resolution. So we have exactly the mass of the compound, and then there are libraries that have these exact masses, so you can relate to the compound.
Okay, so then it's still a tentative identification based on libraries. Can you also use like standards or involve in the quantitation 8-oxo-DG standards in a way, spike the sample or whatever?
Yeah, that to be 100% sure, you mean?
Yes, but
Yeah. Yeah, I think would also be ideal because I analyze in high mass resolution, like it was due to time constraints, but it would have also been great to do MSMS because the fragmentation never lies. So,
Sure.
Yeah, if you really wanted to know if that's the compound, you could have also done MSMS, and then with the fragmentations, then you know for sure.
Whether it's that, because it's really a relevant marker for carcinogenesis, you know. So if you measure that in the zebrafish, I'm beginning a bit worried, but it's also under controlled conditions. We never know whether it's in the environmental settings always the case. But also when I look at the picture, you see then the intestines. But what's the big blur above this picture then?
Could you repeat the page again? Sorry.
152?
Yes. What?
So you see then the intestines. These are colored, and okay, there's then 8-oxo present. But you see also a big blur above in the animal. What's that? Just an open question. I'm not familiar with these pictures.
The blurry?
Yeah. You see also there very yellow at the PS-treated animals. You see also this yellow blur in the animal.
Yeah, but like I mean, we focus on the intestine because it was the main contamination source, like where the microplastics were expected to be more cause and effects. But yeah, we also see that this compound is actually more highlighted in the muscle tissue. But yeah, actually, I didn't do a zoom in. It would have been great because here you just see the entire fish, and we just focus in the gastrointestinal system. So it would have been ideal to do the zoom in. But yeah, what it means that it's also present in the muscle, in the other tissues.
Okay, so maybe that is something for further research. Do I still have some time, Prorector?
Yes.
Okay. So I was also then intrigued by the metabolic changes you are observing. And one of my questions would be, what are the toxic results of these metabolic changes? I mean, do you have any cut-off levels or criteria? Because if you see changes, it doesn't necessarily say that also there are toxic effects. It can be protective effects, it can be adapting metabolism. So have you any ideas about that? Or have you looked at functional issues in this zebrafish? Or how do you know whether these changes are toxic or not? That's the basic question.
So what I did is the compounds that I saw the changes, I saw like what are they related to? So if they were related to oxidative stress or inflammation, and then that's how I related to the toxicology. Yes. But I would say like just changes in few metabolites doesn't mean that much in biology. So I think you should do further pathway analysis. And in MSI, actually, we have a protocol already developed that you use laser microdissection. So you focus on the tissue you are interested. In this case, would be the gastrointestinal system.
Yes.
And then you would do proteomics, and that would be a way to do pathway analysis and more into detail
and then you can
into
see
the toxicology.
You can see. That's I think a really interesting approach to study that. I'm very pleased with your answers. Thank you very much. Give the floor back to the pro-rector.
Thank you very much. Then the opposition will be continued by Professor Van Rooij. He is professor in plasma chemistry at our university, and I'd like to give him the opportunity to discuss with you.
Thank you, Mr. Pro-Rector. Dear candidate, compliments. A sound thesis in the 3 years. I'm very impressed. But let's look also whether we can find some things that we can challenge each other on. Let's start easy with propositions. The last proposition, please.
Oh, sorry. Do or do not, there is no try. Yoda.
It's a good proposition because it triggers many thoughts, and I'm now curious for some of your thoughts. So one of my thoughts was, is this now contemplating, looking at this thesis that is full of tries, and you wish for more? Please enlighten me.
Hi, esteemed opponent. Thank you for your question and your kind words. So I love Star Wars. I wanted to include something related to Star Wars. And my phrase is always, go big or go home. So sometimes you just have to do and like don't overthink that much and just try things. And if you fail, but just do things.
I like that very much. So try or not try. Very good. Now let's go to chapter 2. That brings back sweet memories. And the chairman of the committee has started on chapter 2 already, saying how promising it is. And not to be negative, but I'm going to pester you with trying to find out how much not promising it is. Curious to see your answers here. It's about quantification, and then the first thing that comes into mind is, we often look at species that we add to the system in order to calibrate, and I think you have a perfect internal calibrant in your system, and you have a plot of it. Where is it? It's your isotopic pattern. I cannot find it in me. Page 67. A better calibration you can, of course, not get from nature. The isotopic pattern.
Yeah.
And are you now happy if you look at figure 7?
Sorry, what's the question?
The question is, I see the isotopic pattern as a perfect internal calibrant. Mm-hmm. And I see a theoretical and experimental pattern. Yeah. They are not the same. Mm-hmm. Are you happy or are you not happy?
We were happy because we published this, so I assume we were happy.
Very good.
But
I accept your answer. We go on. I also like very much figure 2. I see the isotopic distributions. And now I start wondering, let's go to the PPG. No, let's go to figure 1. It's larger. And I need my glasses. I see, let's first look at the MALDI spectrum. And I see there are some fluctuations in the peaks at the different molecular weights, at the different polymer lengths. Is this, in your opinion, statistical errors, or is this systematic something? Is this that changes every day that you measure the spectrum, or is this always the same pattern?
You mean that the signal it's different?
Yeah.
Yeah, like with the polymers, you really expect like the Gaussian shape, and we see that for MALDI, yeah, it's not perfect. But yeah, I think that it's from the experimental error. Like, yeah, like it differs day by day with the instrument, so it could be experimental variations. We didn't go much further into trying to know why this happened.
Yeah, because what I find striking then in your MALDI 2, then you get rid of many of these variations, huh?
Mm-hmm. Yeah, it looks nicer.
Yes, that is promising, by the way. Very good. But then I wonder what can be the origin? And one of the things that I was looking at is you have sodium adducts and you have potassium adducts, if I see it right? In between the sodium adducts, you have potassium adduct peaks.
Yeah, well, we labeled mostly the sodium and the protonated, but there was also potassium, but in smaller quantity compared to the sodium.
And what I find striking then is that the potassium distribution, again, it shows a difference.
Yeah. Again, we didn't look into that much, so maybe our fault in this case.
No, I'm not saying that your fault, but I noticed that there I get very excited. You are not really getting excited.
Yeah, because I was like, oh, this we didn't take into consideration.
No, because it's about quantification, eh? So if we do not...
We were mainly focused to know the difference between MALDI 2 and MALDI and trying to know which technique is better. So. Yeah. And of course, we know that it's our polymer because we were just analyzing the standards and the difference between the peaks. It's corresponding to the polymer we are studying. So.
Yeah, but I'll try once more to engage you in some enthusiasm, because what I now, if I would put the knife on your throat, is this varying day by day? Or is this always the same pattern that you measure? What is then your answer?
I would say it can change, but these were like 3 measurements, so we did like the overall spectra. Okay.
Now I hope that you go back to it and check that, because if it's every day the same, then there's maybe some inherent ionization effect that is dependent on the chain length of the polymers. And otherwise, it is a strange matrix.
We didn't.
Effect. But let's go on, because I also like very much the polystyrene actually, where you see that by post-ionization, and I understand your Rampy scheme, and I see also the effect that suddenly you have nothing to do with the matrix anymore in the ionization. You see, I hope at least, I'm challenging, and I'm not saying that what I'm saying is true, so I'm curious for your answer. It's the direct ionization. Do you then really need still a matrix for polystyrene if you are so good in ionizing? Do you also really need it in the desorption step?
So you really need it, because when we didn't use the matrix, in case A, you can see the signal is quite low. So if you compare it to B, when we use the matrix, really good signal. So
maybe use the wrong laser.
Sorry, maybe
you used the wrong laser given having no matrix.
No, I used the good one. I'm sure.
I like that one. But let's make a little bridge to the rest of your thesis, because I also saw your fish, and that's difficult for me to relate to fish. But there I saw that your fish, you cover with matrix. Is it then still a matrix? It's a layer. And what does this layer then do, in your opinion? So you cover your fish under the matrix, under a blanket. What's the matrix doing, and what do you still expect there? And can you then also still relate what you learn in chapter 2 with what you are doing in your fish?
Yeah, like the matrix, what it does is that helps the molecules to get loose from tissue and ionize, so it helps the lipids or metabolites in this case.
Dear Mr. Candidate, you are allowed to finish your answer, but you have the right to say, I'm finished. What do you prefer?
I'm finished.
Okay. That's clear answer. So, Ms. Millán, the time appointed for defending your thesis has officially passed. The degree committee will now withdraw to discuss not so much the quality of your thesis that has been approved already, but in particular the way you have defended your thesis this morning. I request that you and your company await the results of our deliberations and the return to the aula. Thank you very much.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. I know you know that can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Ms. Candidate, dear Ms. Millán, the degree committee here present has assessed the quality of your thesis and in particular the way you have defended your thesis, also taken into account the presentation which you have given this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Compliments. Professor Heeren is authorized to confer upon you this academic distinction in accordance to Dutch university customs, and therefore I invite your supervisor now to take the floor officially.
All right, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Lidia Molina Millán, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dear Dr. Molina Millán, dear Lidia, congratulations, you did it. You are standing here now as Dr. Molina Millán with your diploma in hand, less than 4 years after you started in Maastricht in October 2021. So you completed this journey in 3 years, a little bit more than 3 years, which is impressive. It's an amazing achievement. Every PhD is a unique journey filled with unexpected events, new findings, challenges, new encounters, disappointments, successes, struggles, aha moments, insights, discoveries, successes, struggles, interactions, travel. And your journey has all these elements incorporated in it. Importantly, they all contain many celebrations, but the most important one is the one after the final defense today, because you did it. So go big or go home, you went big, you did it. Today is also an interestingly unusual day to defend, as Maastricht University is or will be on strike in our fight against the government perception that science and education budget can be significantly reduced. Moreover, Maastricht's international signature is under political pressure as well. A strike. While here, we celebrate your outstanding achievements that are exactly a testimony of why it is so important to keep on investing in talented researchers such as you, Lidia. You came to us with a master's degree in analytical chemistry in Spain, accompanied with a drive and passion to make an impact in understanding the effects of plastic pollution in aquatic environments. As we just had received this European Union Marie Sklodowska-Curie grant, Futranet, spearheaded by Neves Ogrinch, online here today, it was a perfect fit. And already then you impressed us with your presentation qualities that we were able to witness here today and that the entire committee was very much impressed with. Together with your supervisory team, Eva Kuipers and Berta Sierra-Pastor, we started this journey towards that piece of paper in the back there. And it did have all the elements that I described earlier. It was a roller coaster ride filled with highs and lows, sharp turns of directions, uncooperative instruments, and rapid acceleration where needed. Actually, being here together today is extra exciting, as you yourself have questioned many times if you would be possible to complete a PhD in the Netherlands in 3 years. Look at that, it worked, you did it, and that was you. While we were convinced you could do it, if you put your mind to it, you were struggling with the many uncertainties life was throwing at you. But in the final year, this big acceleration kicked in. And I don't know what happened, but you really impressed all of us with how you pushed through and managed to complete a thesis where almost all of these chapters are now published and resulted in the success of today. And you've truly found your independence as a researcher, an objective we discussed on day one of your PhD, where why do we want to do a PhD? Because you want to be an independent researcher, and look at you here today, impressive. Your personal journey has truly been a journey of exponential personal and professional growth in challenging times, and I do want to compliment you for that, because it was not always easy, but you did it. You dove deep into the fundamentals of polymer science, together with Alosha, Johan, and Chris Westdemiotis, who joined us. Chris joined us on a sabbatical visit. I found it amazing to see how quickly you picked up analytical polymer mass spectrometry and polymer and spectral interpretation. When we had a discussion with the committee, and I'm not supposed to disclose any detail, but everybody was impressed of the breadth of your knowledge, all of these different elements that you brought together. Pretty good. Something I think you did not expect as we started on this trip. Talking about trips, we all have preferences when we travel. And I think your preference was anything and everything Spanish. True to your cultural heritage, collaborating with the Spanish group of Laia Navarra team, Joachim and Albert, who's here in the audience today, a Spanish co-supervisor, a committee chair with Spanish links, lots of Spanish talk in the hallway and Spanish food during different celebrations, and lots of wine along the way. A secondment to the IDEA CSIC lab, part of the MSSC, the Marie Skłodowska-Curie experience, even allowed you to spend some time during your PhD in Spain. But you traveled beyond Spain and the Netherlands. Futronet brought you to different summer schools where you had lots of fun with the team, workshops and conferences across Europe, where you communicated your science, and you're an outstanding communicator, and that was evident again today. You became one of the social media PR representatives of Futronet, the ERC representative of Futronet, the early career researcher representative, showing that you also developed many other skills to become this all-round responsible scientist. You're also a very social person, and we're driving forward many social events in the lab to the extent that we sometimes thought you liked the other things more than the actual PhD research. When you travel, you need to stay balanced, and you always manage to find the right balance for you. In the latter part of the PhD, you were even involved in starting up the Young MVMS, where you helped organize young mass spectrometrists in the Netherlands to build professional careers, where initially you put mental health and well-being on the agenda. And I think that's an area where sometimes in times of high pressure during a PhD, we need to put more emphasis on this, and you made us very aware of that. Your trip here at MFRI has now come to an end, and you are exploring your next destination. Wherever this will take you, I am convinced that you will continue to grow and make an impact on the world you shape. Let your passion for science guide you, as it will bring you to many unique, unexplored places. Let us all make sure that such passion for science gets full support for all of its needs, financially, politically, and societally. Only then we will keep educating young, talented, young doctors like you, Lidia. I want to end with congratulating you once more, and of course I want to include your family in these congratulations. It was a true pleasure supervising you, and I could not be prouder of what you've achieved today. Thank you for everything that you've brought to the group, and now it's time to celebrate. Congratulations.
Dear Dr. Molina Millán, how does that feel? Feels good?
Amazing.
Amazing, that's exactly the right term. Also on behalf of Maastricht University and the board of directors, I really would like to congratulate you with this highest degree which you can achieve here in our country, in the Netherlands. There are two remarks I would like to make. First of all, you did a lot of work also in the Corona time. Nobody has mentioned this, but you started almost at the end of Corona. And basically, what I noticed in your thesis is that you had to do a lot of practical work, because a lot of theses in Corona were reviewing, write a review, to stay home at your computer, but you work a lot here. And secondly, I got your thesis about 2 weeks ago. I was very disappointed. You know why? About the front cover. Because you cut your zebrafish in two. And the most important part of the zebrafish is, of course, the brain. And you took out the brain, and I thought, why is she doing this? And listening to your presentations, in your slide 12, you can check later, you can see the accumulation in your intestine, but there's much higher accumulation in the brain. I saw it on your picture over there. So basically, I think there's room for a further study and really look to how these plastics go into the brain, and that's really something which is important. But saying this, it is already mentioned by the first promoter, by Professor Heren. We as a team here, and this I as the pro-rector, we came to a unanimous positive decision, so we were very pleased to be work at this. Anyway, in my congratulations, I also would like to go to the promotion team. They guided you, and I think it should be well respected that they guided you in such a short time, in 3 years, to do this. I also would like to thank the members of the Corona to join here this morning, also Professor Oknis from Slovenia, and they had to read your thesis, prepare questions, ask questions, etc. So that's an important thing too, and I would like to thank them. And in addition, my congratulations goes to your family here on the front line. I was tempting to speak some Spanish, but I decided not to do this. Also, I also have some family in Spain, to my surprise as well. And again, to the students in the audience, you can hear they are sitting a little bit in the back side. I hope all of you to see in one of the forthcoming PhD theses, and it was a very nice thesis. And by saying this, I'm going to close officially the session using my hammer, and this session is now closed. How do we proceed? The members, we here in the Corona, the new young doctor, we're going to make a picture here with the online presence of Professor Oknik. Then we make a picture on the stairs, and you in the audience can already go to the rafter and start what was mentioned. Have a glass of wine, maybe some Spanish wine, or take a coffee or whatever you want, and we will join you in about 8 minutes. Thank you very much.